Hi everyone, welcome to Zyphie's study channel. Today I'm going to review the steps and process of the metabolism of nucleotides. This video serves as an educational video and also for my own personal revision, and I hope it will help you. So first, let's talk about the structures of bases, nucleosides, and nucleotides. I'm not gonna draw the whole whole structure of the bases, but I'm going to tell you some tricks on how to identify different pyrimidines and purines, and the difference between nucleoside and nucleotide. So first, with the bases, we have pyrimidine, which is cytosine, uracil, and thymine. And if you look at the 12 o'clock, well, let me draw a clock here. You can find an amino group, a carboxy group, attached with a methyl group. So here you can tell that in order to form thymine from uracil, you need to add a methyl group. And we will talk about this later in the process of de novo synthesis of pyrimidine. And now with purine, also 12 o'clock, but we all know it has 2 rings, right? So in the second ring, adenine has an amino group. And then guanine has a carboxy group. So now the difference between nucleoside and nucleotide. So here, nucleoside, it's base plus pentose sugar, which are ribose, or deoxyribose. But then nucleotide is nucleoside plus a phosphate group. So here's how you identify and differentiate between different bases, nucleoside, and nucleotide. And now let's move on to the 2 pathways we can use to synthesize nucleotide pathways. First, it's called salvage pathway. Here we use activated ribose, which is PRPP. Later we will talk about how PRPP is formed and its full name. And we add a base, and this base is reutilized. Now, on the second pathway, we have de novo, which means from scratch, or from basically de novo, from new. We also have activated ribose, PRPP. We have amino acids, which can give us amino groups, carboxyl groups, and other carbons, which are really important, and we will talk about that later. We also need ATP because we need to add those amino acids onto the ring. And then we have carbon dioxide. Well, basically bicarbonate. And then they give us nucleotides. Nucleotide. So now, first, we'll talk about the synthesis of PRPP. So what does PRPP stands for? PRPP means phosphoribosyl pyrophosphate. And normally we have on the fifth, the one. And the fifth is the phosphate group. By forming PRPP, we need to have ribose 5-phosphate. And where does this ribose 5-phosphate comes from? Of course, we can remember the pentose pathway. So from ribose 5-phosphate, we need ATP to give us 2 phosphate group to the ribose 5-phosphate. So we need this 2 phosphate group to form PRPP. Which is added onto the first carbon ring. And as this process synthesizes PRPP, so a very easy name to name this enzyme is PRPP synthetase. So now, let's move on to pyrimidine synthesis. De novo. So the basic, like a general view of this process is start with bicarbonate. An amino group. And then we caused 2 ATP to form carbamoyl phosphate. And then we add in an aspartate to form a pyrimidine ring. And then through several processes, let me try better. We combine it with PRPP to form UTP. And you know, it's through adding an amino group to form cytosine T triphosphate. And then here you can form TMP, and CTP can form deoxy. CTP uses an enzyme that's really important, and we will talk about later. And then this two can be used to form RNA, this two to form DNA. Now, let's get into the detail of de novo synthesis of pyrimidine. First, we have bicarbonate, right? And we need to transform it into carboxy phosphate. Yeah. Well, how do we transform that into carboxy phosphate? Well, you guessed it. It's by adding a phosphate group. And where do you add a phosphate group? Of course, form ATP. This is a reversed reaction, just to make sure. And then from this carboxy phosphate, we transform it into carbamic acid. What a weird name. Well, how do you transform into carbamic acid? As we all have the carboxy group part. So then we need to add an amino group. To form a carbamic acid, the amic. And where did this amino group come from? Well, of course, our dear friend, glutamine. Be careful, it's glutamine. You hydrolyze it to give this amino group. So glutamine will become glutamate, which is glutamic acid. So now we have carbamic acid. We add another phosphate group. I'm not going to draw the detailed molecules because it kind of takes up a lot of time. And you can all search it up on internet. So here, by adding a phosphate group, what do we do? Again, as always, ATP. ATP always gives us phosphate groups. Here, I didn't write any enzyme names. You know why? It's because we only have one enzyme. To perform this process. It's called CPS2 enzyme. And the full name is, you guessed it, of course, we're making carbomoy phosphate. What else could you call it? It's carbamoid. Why I always pronounce it wrong? Carbamoid phosphate synthesis synthetase. It's reading difficult to say. Synthetase. And the structure. I think you can find it in other textbooks, such as Biochemistry from Freynmann. And then you have this enzyme, as there are 3 reactions taking place in one enzyme. So of course you have 3 dominions. 3 domains. Oh, what am I doing? 3 domains. Sorry guys. That's a multilingual problem. So here you enters. Well, here it goes glutamine, and then it turns to NH3, and you form carbamic acid. And lastly, we have carbamoid phosphate. Honestly, I really shortened down the processes, and compared to the class presentation I had, was way too complicated. And this is very simple, very easy to understand, but in one condition. It's only for revision. Better go to class or listen to other videos that are more detailed, but I think this really helps with revision. I'm having my exams in about 1 month. Well, 20 something days. So I think making this video really helps me to revise, and also helps you to revise. So now let's move on from carbamoid phosphate. Well, remember we mentioned this part where we add an aspartate to carbamoid phosphate to form a pyrimidine ring. So now this is what we're gonna do. We add aspartate. No surprise, aspartate. Right. We replace the phosphate group, and now we form a molecule called dihydroorotate. Oh, I always call it orate somehow. Should be correct. Or rotate. Oh wait, went too fast. Went to another process. I mean, you add an aspartate, of course it's called carbamoid aspartate. Sorry guys. And because you're adding, you transfer a aspartate to replace the phosphate group. Then the enzyme, it's called aspartate carbamoid transferase. Now let's get to dehydroorotate. Remember, guys, dehydroorotate is an enclosed cyclic molecule. And then carbomoyl aspartate is not an enclosed one. So now I have to close this circle by getting rid of a molecule of water to form dihydroorotate. And because we're forming the dihydroorotate, so the enzyme is dehydroorotate. The A's coming in. Of course, an enzyme. A's. Finally, we got to orotate. Well, previous name has dehydro, which means we're gonna oxidize it. Well, how do we oxidize it? Something's gonna reduce, right? So we have NAD. NAD is reduced from NADH. And then dehydroorotate is oxidized. Redox. Dehydroorotate. Dehydrogenation. Of course, every time you have an NAD plus to form an NADH, what can go wrong if you name it dehydrogenase? Dehydrogenase, guys. Dehydrogenase. Now, let's talk about something even more interesting. You think CPS2 is another enzymes, and these are some other enzymes, right? Well, all these steps are on the same polypeptide chain. Remember, same polypeptide chain. They are not individual polypeptide chains. They're just one cadence of polypeptide chain. So guys, we're already done till here. So what's next? Well, you can see here, PRPP, the MVP of nucleotide synthesis. We have orotate that we just formed. We add PRPP. Not me referencing to pineapple apple pin. Here, let's be serious. Here, we get rid of the 2 phosphate groups from PRPP so that we can join orotate with the ribose sugar from PRPP. So basically it's PR. Well, no, it's pyrophosphate. Ribose pyrophosphate. So that we have a rotidylate. Oh, why is the name so hard to pronounce? And because we're transferring phosphoribosylguys, phosphoribosylguys, I just said it wrong. We're joining orotate and phosphoribosyl. So that's why I need to get rid of P's. 2 P's. Right. So the enzyme is called arotate phosphoribosyl transferase. Long name. Long names. But what's essential is that you remember what groups you transfer to other molecules. Then you don't have to memorize enzymes. Instead, think about it and name it logically. That's what our teacher always says. Now you have one up. We're here. We're somewhere here. What do we do now? We form UTP, guys, UTP. But first, we need to form UMP. Don't rush. UMP first. So how do you form UMP? Well, you get rid of a carbon dioxide. And what's removing a carbon dioxide called? Decarboxylation. So of course, it's decarboxylase. Decarboxylase. And you know where the carbon dioxide is removed? It's on the 5th phosphate. 5th. Carbon. So then it's called orotidine 5' phosphate. Oh, no space. Decarboxylase. Haha, now. What we have here? UMP. Alright, UMP. So what do we do with UMP? Of course, we're going to transform it into UTP. But first, we need to give it a extra phosphate group to form UDP because it has 2 phosphate groups. What do we do? Well, as usual, our old friend ATP. And here we use a kinase. MP kinase. Let's change the color for enzymes. Don't we love colors? And now we use another kinase to form UTP, of course, old friend of mine. ATP is our old friend. Nucleosid diphosphate. Oh, not in Catalan. Kinase. And now, let's get to the fun part. If you guys still remember what I just said at the beginning of the video, uracil, it's CO group here, right? And you have amino group on cytosine. So what do we do? Amination. You give an amino group. So let's move on. UTP 2 CTP. But how do you give it an amino group? Of course, another old friend of ours for synthesis of nucleotides. Glutamine. let me re-emphasize this. It's glutamine, not glutamic acid, not glutamate. Okay, it's glutamine. And don't fall for that in your multiple choice questions. Be careful, because that's how I paid my lesson. You hydrolyze it. That's the byproduct. You get rid of now. Of course, there's a cost. I don't know, friend. Now you form CTP. Well, guess what's the enzyme called? Really easy. Easy peasy. Lemon squeezy. Of course it's called CTP synthetase. Man, you have an ATP. Of course it's synthetase. Hello. Fun part now. Regulations. Let me change the color. Regulations. I bet your professor always give you a question on regulation. Why wouldn't the professor give that to you? Why wouldn't? You know how it forms? 5 steps. CTP. So, high concentration of CTP. Heya. You know what enzyme it inhibits? Which enzymes? Aspartate carbonyl transferase. And that's the only important part of the regulations of pyrimidine de novo synthesis. Now, let's get to the salvage part. Here are some formulas. A general formula to remember. Base. Yes, base. Cytosine, uracil, Pyrimidine. And then you have ribose, 1-phosphate. You form. What do you form? Nucleoside. Good. And what's the enzyme called? Nucleoside phosphorylase. Because you're getting rid of that phosphate group. On the second part, you have nucleoside. To emphasize that, right? Side. You add our own friend ATP. What do you get? Your nucleotide. Nucleotide. Mono. phosphate. Nucleotide monophosphate. ADP for sure. And what's the enzyme called? Nucleoside kinase. Wow. Of course. Given a phosphate group. Let's have an example. Give an example. Thymine, right? Thymine. Example. Plus. Why not? Do you guys prefer deoxyribose or ribose? Nah, let's go with a ribose. 1 phosphate. What'd you get? A nucleoside. And that's Pyrimidine. And what's the enzyme called? Thymidine phosphorylase. Now we have thymidine, the nucleoside, ATP. Oh, I really don't know how many times I said ATP. We'll count that in the video. TMP. What is it called, TMP? Thymidylate monophosphate. To no surprise, thymidine kinase. So guys, here's the first video on metabolism of nucleotides, which is here in which we mentioned how to identify structures of pyrimidine, purine, nucleotide. And then we mentioned 2 pathways. Ooh, that's bright. And then we talk about the synthesis of BRPP. Super duper important in the next video. And then we mentioned the general diagram of pyrimidine synthesis from de novo. And then we talked about the different processes, enzymes, and regulations. That's the only regulation to remember. And then how we transform UTP to CTP. Easy peasy. How you remember it? From the difference of structure. Ooh, I think, okay. I think that's all. If I forgot to mention anything, please comment down below and then give me some suggestions, as this is my first ever video I actually published. Amazing, right? I just wanted to share how I study. Well, basically, making these videos is just to help me to not watch any other videos, to waste my time, and actually concentrate on my study. And I think making videos really help, really boost my memorization of content or information that I need. And I remember it for my exams, and making videos won't make contents like making videos won't make my information that much boring at all. I think I really like it, just talking to you guys. What's it called? The Freynmann Technique, where it teaches other people. My teacher always, well, I think my high school teacher always stressed that the best technique is to teach someone else. But since if I teach my parents or my friends about the content that I'm studying, they would never even understand what I'm saying. So the best way is to make videos and share with you guys, so we can all improve and can all enjoy our study. Thank you guys. Hope you really like this video. Give a thumbs up and then subscribe to the channel. See you next.